It's Nelson Freimer, and I am just really, really delighted to introduce our speaker today, Hopi Hoekstra from Harvard. As she knows, and as some people here know, we've been trying to get her to give us a seminar for a few years. We really thought that we were going to be at the point now where this would be in person, but you know, obviously we're still remote, and hopefully we'll be able to get her to come out again sometime in the not that distant future so people can meet her in person. But today she's going to be talking about how natural behavior evolves, lessons from wild mice, and it really speaks, I think, to really a field which, where I have to say, in my view, she really has no peers. She trained in evolutionary biology. She had a Ph.D. in zoology from the University of Washington, then did a postdoc in ecology and evolutionary biology at University of Arizona, joined the faculty at UCSD before moving to Harvard, where she is now the Alexander Agassiz Professor of Zoology, and is also an investigator of the HHMI and an investigator at the Broad Institute. And again, what I think is really extraordinary, her lab works on genetic basis of morphologic and behavioral adaptation of wild rodents, as you will hear about. And if any of you look at her website, which I really would encourage people to do, she divides up the work in her lab in several different areas: population genetics, evolution and development, behavioral genetics, sexual selection, and experimental genomics. And what's really extraordinary is if you look at any one of those areas, you would think this is somebody who has an entire career in this one area. And then you realize basically they're doing this in these 5 different areas and putting it together. And I really don't understand how that's possible, but hopefully today we'll get at least some sense of how she does it. So Hopi, start.
Great. Thank you so much for that introduction. And these gray hairs are in part due to trying to balance all of these different research areas at once. But for me, that's what makes this process so fun. It's being able to sort of traverse fields and let the science lead you in different directions. Okay, how is my screen? Or am I not sharing? Let's see. I've got 2 different screens up here, so give me 1 second. See how this works. There we go. How's that? Perfect. Okay. So thank you very much for the invitation. I too am disappointed I won't be there, but I have a sabbatical starting in approximately 45 days, and I hope to spend some time next spring in California. So maybe I can meet folks in person then. So today what I'd like to do is tell you a story about how at least one type of natural behavior evolves. I had a very difficult time thinking about what I might talk about today, so I decided to talk about one complete story to give you a flavor of the type of work that's going on in our lab. So let me start by motivating this work with the question that really encompasses all the work that we're doing in the lab, and that's this one. What are the genetic changes that are responsible for variation that occurs in natural populations? I'm trained as a geneticist, and so we spend a lot of time trying to identify genes, and in some cases even the mutations that are responsible for variation, and in particular variation that we think matters to the organisms in their natural habitat, that affects their ability to survive and or survive to reproduce. Now, for many geneticists, finding a gene or a mutation represents the end game. But for us, it really represents the beginning, because armed with the gene in hand, we can then start to ask, in my opinion, the very interesting questions about how this trait evolved. What are the evolutionary processes responsible for generating this variation? And on the other end of the spectrum, the precise molecular mechanisms. How does a single change in a protein or in a regulatory region act through development to actually produce the variation that we see in the wild? So in other words, we can understand both how and why populations adapt to their local environments. Now, the goal of trying to connect genotype and phenotype is not new. Of course, for decades, biomedical geneticists have been trying to identify genes and mutations that contribute to disease. This is a sort of first step in developing therapies or diagnostics, for example. We work to address the same question, but I like to think of it as sort of the happy flip side of the coin, in the sense that we're looking for genes and mutations that are beneficial to individuals in natural populations. And I would argue over the last several decades, there have been a growing number of examples in which the connection between genotype and phenotype have been made for a diversity of organisms, in particular focused on differences in morphology. So many of you are probably familiar with the elegant work stemming from David Kingsley and Katie Peikel's lab, looking at, for example, the evolution of this bony armor and these spines on the back of stickleback fish, identifying the molecular mechanisms driving morphological differences in this armor, depending on whether fish occur in marine or lake environments. On the other end of the spectrum, there are these beautiful mimetic color patterns that are seen in heliconius butterflies, which we now have a strong understanding of the genes involved in generating those patterns. Our lab too is contributing to this growing literature. We started the lab focused on understanding the genetic basis of adaptive pigmentation, working with these mice that I'm going to introduce you to later. So we've spent many years trying to identify genes, and in some cases even the precise mutations that contribute to differences in color and pattern, and how that affects the ability of these mice to survive, where the coat color matches their local environment and protects them from visually hunting predators. Sort of bolstered by the success of being able to connect genotype and phenotype for simple morphological traits like pigmentation, we got excited about extending this approach to more complex traits. So we focused on reproductive traits and have been able to identify a gene that contributes to differences in sperm morphology that affects their the ability or the speed at which sperm swim and their competitive ability in the canonical race to the egg. And then we've gotten even more complex in terms of the morphological traits we've been looking at, most recently looking at skeletal traits. And in particular, as I hope you can see in these x-rays, differences in tail length that are associated with the environments that these mice live in. Long-tailed mice are found in forested habitats, where longer tails aid in climbing in trees compared to their prairie ancestors. So from this work, we've been able to learn several lessons. So the first thing that this nicely demonstrates is that even in non-model species, we're at the point where we have the tools and approaches where we can really identify genes and mutations and use them to understand both evolutionary and molecular mechanisms driving variation in natural populations. But one important thing to keep in mind as I tell you this story is that when we first started this work with morphology, the first studies really focused on known genes. So for example, when we were studying pigmentation, some of the first links between genes and phenotype, in this case coloration, involved genes like the melanocortin-1 receptor or the agouti signaling protein that had long been studied by mouse geneticists and known to be important in pigmentation. But over time, as we dug deeper, we identified genes like corin that had not previously been implicated in color differences, or even genes like ALX3 that hadn't even been implicated in pigmentation. So while the first studies really focused on candidate genes, eventually we were able to discover novel genes and novel mechanisms for natural variation. And that's really the point we're trying to get to. So about maybe 10 or so years ago, we asked ourselves, okay, we have a good handle on approaches that are helping us make this connection between genotype and phenotype for morphological variation. Can we apply this same approach to understanding variation in behavior? And I have to admit, I was quite nervous about this. And I'm sure many of you who study behavior also recognize that there are challenges to studying behavior that just aren't as big when we're studying morphological variation. So for example, Measuring behavior in many cases is more complicated than measuring, for example, coat color or length of the tail. Of course, we've made great strides in recent years with new machine learning-based approaches to quantifying behavioral variation, but nonetheless, it still remains a big challenge. Second, we now have to deal with what's arguably the most complex and heterogeneous tissue in our body, the brain. And then third, again, relative to morphological traits, there's a sense that behavioral traits overall had lower heritability because they may be more affected by environmental stimuli. Hence, we had lower power to make the connection to genotype and phenotype, although it's unclear if that's actually true. So we asked ourselves, okay, this is going to be harder to study behavior compared to morphology, but could we offset some of these negatives or these challenges by capitalizing on really large and heritable differences in behavior that have evolved in natural populations? Well, we convinced ourselves it was a good idea to give it a try. So today I want to tell you a story about our attempt to make the connection between genes and behavior by telling you a story that involves social behavior. In particular, we got excited about social behavior because, of course, it's a hallmark of human society, and dysfunction in social behavior can have major impacts on human health and well-being. Moreover, there's a long history of studying social behavior in the context of evolutionary biology in a number of different organisms, from social amoeba to ants and honeybees to cooperative behavior in vertebrates like fish and birds. And one interesting thing that struck me was that a postdoc in my lab, Sarah Coker, who's now an assistant professor at Princeton, started to work on sweat bees. And she was interested in sweat bees because there were some populations within the single species that were solitary and others that were highly social. And using a whole genome association study approach, she was able to implicate a gene called syntaxin-1 in contributing to differences in sociality among these populations of bees. And what was so striking about this result is that syntaxin-1 is a gene that's been previously implicated in autism spectrum disorders in humans, which is a study along with several others that suggests that the genes that contribute to social behaviors may be conserved across great evolutionary distances, so from bees all the way up to humans. But by and large, of course, most work on the genetics of social behavior has arguably been done in mice. And lots of beautiful results have come out of this work, in large part because of the amazing tools that we have to interrogate the genomes of these laboratory model systems. But like any system, they too have their limitations. And one of the things that's challenging, in my opinion, about studying social behavior in mice is that the differences in social behavior, even among divergent strains of mice, is limited, at least relative to other species. And this is certainly what contributed to folks like Tom Insel and Larry Young in developing a complementary system in voles. In voles, differences between monogamous and promiscuous species have contributed to large differences in social behavior between species. But this system has, using a candidate gene approach, the system has really implicated the vasopressin and oxytocin pathways, and in particular mutations in the vasopressin receptor and oxytocin receptors being really important in contributing to differences in social behavior in these rodents. But again, like all systems, voles too have their limitations, and that's that these species that are the focal species in this system, while both voles are divergent enough that they're not interfertile, and hence you can't do genetics. And that led us to think that one of the groups of mice that we've been studying in the lab might be an additional complementary system in which we could contribute to this growing literature. So let me now introduce you to the main actor in today's talk. my favorite group of mice. These are deer mice in the genus Perimiscus. Now, to many of you, these mice may look like a typical mouse, but perhaps surprising is they fall well outside of the split between our standard laboratory organisms of at least mammalian organisms, mouse and rat, which let's say are separated by about 10 million years. Perimiscus are about 25 million years diverged from mouse and rat. So this is a nice evolutionary position in the sense that they're close enough to mouse and rat that we can borrow and steal those wonderful tools that have been developed in these model systems, but divergent enough that we may learn something new by studying Perimiscus that we may not learn by just focusing on mouse and rat. In addition, Perimiscus is a genus that's quite speciose. Depending on who you talk to, there are about 55 species of Perimiscus. So not only are we able to make comparisons between Perimiscus and mouse and rat, but we can also take advantage of variation within this genus and compare more closely related and sometimes interfertile species. So Perimiscus-like laboratory mice can be reared in captivity in nicely controlled environments. And though we're quite far behind, our lab and others spent considerable effort developing molecular, genetic, and genomic resources for these mice. So we have reference genomes, we have lots of population resequencing data, we're using viral vectors to trace neural circuits, and we have transgenesis working, although not as efficient as we'd like. But here's where Perimiscus differ from laboratory organisms, laboratory mice, or their wild counterparts, which we often refer to as house mice, in that they're not human commensals. Instead, they live in natural habitats throughout North America, where they have a very wide distribution. So this image just shows you the species range for a single species of Perimiscus. And in their habitat, they're often really highly abundant. In fact, the most abundant mammal in North America. And because they can be found at all sorts of habitat types, from the sort of cornfields of Nebraska to the top of the Rockies to islands off the coast of Maine, there's a lot of potential or opportunity for local adaptation, which is certainly contributing to the great variation in both morphology and behavior that we see both within and between species. So we can really capitalize on this natural variation. Now, in terms of behavioral diversity, one of the first and most striking examples of diversity within Perimiscus is their diversity in their mating systems. So here I'm showing you a phylogeny and the relationships among 7 species of Perimiscus. So these are not all 55 species, obviously, but are representative of all the major lineages. And what you'll notice is that, like most mammals, most Perimiscus are promiscuous, as indicated by this orange color. But what's really interesting in this group is, like voles, it looks like monogamy has evolved multiple times independently, at least twice independently in 2 different parts of this tree. This allows us to look at convergent or parallel evolution. But today I'm going to focus primarily on the comparison between these 2 species, Perimiscus polyunos and maniculatus. And although they're closely related sister species, they're known to have highly divergent mating systems. So Perimiscus maniculatus is a typical promiscuous rodent where males and females seem to mate with basically whoever they run into. So in other words, if you take a litter of mice from the wild and you genotype that litter, more often than not, that litter is sired by multiple males. By contrast, Perimiscus polyunos is both socially and genetically monogamous. So if you genotype a litter, it's always sired by one male, and it's the male that's interacting with that female. But because these mice are sister species, if you bring them into the lab and don't give them any choice, they happily will mate and produce viable and fertile offspring, opening up the opportunity to do genetics. So this observation that these 2 species have divergent mating systems got us excited about differences in parental behavior. And that's because parental care is thought to have evolved in monogamous species, as males in those species are more certain of paternity. So there's a correlation between parental behavior and mating system. And this raised the question of these two species that have different mating systems, do they also have differences in parental care? So to test that hypothesis, my postdoc Andres Bendeski designed a simple behavioral assay, which allowed us to quantify different components of parental care. So let me explain the assay. So what we did is we took a male and a female, put them together, they mate, and when they have offspring, we tested them at 3 time points when the pups were 3, 4, and 5 days old. And the testing went something like this. We could take independently the male or the female out of its home cage, introduce it to a new cage with nesting material, and allowed it to acclimate to that cage for 30 minutes. Then after 30 minutes, we introduced one of its own pups into the cage, and we monitored its interaction with that pup for 20 minutes. Then after 20 minutes, in 2-minute intervals, we introduced the remaining pups from that same litter. And in doing this, we were able to quantify different components of parental care, including nest building behavior, the time to approach, handle, huddle, and lick the pups, as well as the fraction of pups that were retrieved from the litter back to the nest area. And again, we did this for both moms and dads, and for each one at 3 time points when the pups were 3, 4, and 5 days old. So next, what I want to do is to show you 2 videos that will give you a sense of the differences and how big the differences are between the parental behaviors of these 2 species. So we'll start off with Perimiscus Polyunos, and that I'm showing you here highlighted in green. And for the rest of the talk, I'm going to associate data in green is going to be from the monogamous species, and in orange from the promiscuous species. So what you can see here is, and both of these happen to be males. So here we see a male in the corner. You can see during its acclimation period, it's made this nice little nest. And so after 30 minutes, we're now going to introduce one of its pups into the cage. The researcher is going to step back, and you can see almost immediately the dad runs over to the pup, interacts with the pup, handles the pup. And while he does go back into the corner to take a little break, he thinks better of it, returns back to the pup, handles it, retrieves it back to the nest, and will spend most of the remaining 20 or so minutes interacting with that pup in some way. So this is what we sort of casually call a good dad. By contrast, Maniculatus, you can see in 30 minutes, hasn't touched its little nestlet, so hasn't done any nest building behavior. The pup, when introduced into the cage, you can see it's probably emitting both ultrasonic and audible vocalizations. The father is hesitant, eventually goes over and interacts with the pup, but retreats back to his corner. And while he'll explore the pup briefly a few more times, we'll spend most of the rest of the assay in the corner, actively avoiding the pup. So this is what we, again, very casually call a bad dad. So we can use this assay then to start to quantify differences in parental behavior. And so let me show you some of those data. So again, monogamous in green and promiscuous in orange. So the first thing we'll look at is nest quality. And as those videos suggested, the monogamous parents do quite well. They build generally a nice nest. And this is in stark contrast to the promiscuous moms and dads who don't do any nest building at all. So this is a clear difference between the 2 species. We see a similar pattern when we consider, for example, minutes spent licking the pups. Again, the monogamous moms and dads do very well, spend a lot of time licking their pups compared to the promiscuous parents. Again, this difference between the species. For some behaviors, we see a difference between the sexes. So when we think about pup retrieval, moms of both species do better than dads of both species. But most often we see an interaction between species and sex. So for example, when we think about minutes spent huddling, the monogamous moms and dads do a really good job huddling their pups, spending the majority of time huddling their pups compared to the promiscuous species. But in the promiscuous species, moms do a better job than dads. So in other words, the biggest difference in parental behavior occurs between the fathers, as one would predict. So overall, when we look at this data as a whole, we see several patterns. So in general, as one might predict, monogamous mice are more parental. And fathers contribute as much, sometimes even more, I loathe to say, than mothers, compared to promiscuous mice, which are less parental, with fathers generally providing less care than mothers. So this is really exciting data to us because it suggests not only is there a correlation between mating system and parental behaviors, but the differences in parental behaviors between these two closely related species are actually quite large. So as a geneticist, of course, I was really interested in the question of, are these differences heritable differences? So one way to get at that was to do a simple cross-fostering experiment. So what we could do was take litters that were born at approximately the same time between a promiscuous and a monogamous parents. And then we'd sneak into the mouse house and quickly swap the litters between the parents. So we now have promiscuous mice being raised by monogamous parents and vice versa. Then we let those pups grow up, pair them, allow them to mate, and then record their parental behavior towards their pups using the same assay that I told you about before. And then we can compare these to mice that were not cross-fostered. I'm not going to go into the details of the data because the pattern is quite clear. We didn't find any significant difference between pups raised by their own parents and pups that were cross-fostered by parents of the other species. So in other words, the parental care that an individual received does not influence the care that it gives to its offspring when it's a parent. And this was actually quite surprising to us that this was such a clear pattern, because studies in rats suggest that the care you receive as a pup really does influence your parental behavior towards your pups. But this is really exciting because as a geneticist, this was consistent with there being a strong genetic component. And that then led us to the next experiment, which was to cross these two species of mice and try to look at the genetic architecture underlying these differences in heritable parental behavior, the heritable component. So we took moms of Maniculatus, crossed them to dads of Polyunos, generated our F1 hybrids. Those F1 hybrids, of course, have a chromosome from each of the two parents. And then we intercross these F2s to F1s to generate a large genetically heterogeneous population of these second generation hybrids, about 800 mice in total. And just by way of reminder, each one of these F2 hybrids has a different combination of alleles from the monogamous and promiscuous parent. So with these mice, we were able to then ask first about correlations in parental behavior when we genetically shuffle these two species. So first, I want to show you the correlations among the parental behaviors in these F2 hybrids, these genetically shuffled F2 hybrids. So I'm going to focus on 6 behaviors that we felt like were easiest to measure, or we can measure most accurately, and that showed large differences between the species. So they're labeled here. So huddling time, licking time, fraction of pups retrieved, latency to handle and approach, and nest quality. I'm showing you the data separately for moms and dads. And the strength of the correlation is indicated by the color of the box. Each box is symmetrical over the horizontal. Okay, there's several results to note. The first one is the patterns are similar between fathers and mothers, but they're not identical. The second is that all the correlations are positive. So if you're good at huddling, you tend to be good at licking. And there's no evidence for trade-offs between these behaviors. Third, some behaviors tended to show relatively strong correlations, suggesting that they act as sort of a behavioral module, such that if you're good at huddling, you're good at licking, and you're good at retrieving your pups. But by contrast, some other behaviors, like nest building behavior, seem to be largely either weakly or uncorrelated with other aspects of parental behavior, suggesting that in some cases, there are genetic variants that may affect multiple traits like huddling, licking, and retrieving, whereas other genetic variants seem to have very specific effects on just some aspects of parental behavior. Okay, so now that we've generated this large population of F2s, we've measured all their parental behaviors in all 800 or so mice. The next thing we were interested in doing is asking, are there correlations between genotypes in these F2 mice across their genomes and their parental behavior? In other words, trying to localize variants that affect the behaviors of interest. So to do that, we used a forward genetic approach called QTL mapping. I know for some of you who are expert, we need not review this, but to make sure that we're all on the same page, I'm going to see if I can get the lights on in my office again. There we go. I'm being too sedentary. So in this case, what we do is we take those mice that our F2 mice, we've measured their behavior, and now we're going to genotype them at hundreds of thousands of markers across the genome using an approach called DD-Rad-Seq, which we developed in our lab. This allows us to do that quite efficiently and cost effectively. And so what we're doing in doing so is what we're looking at is, as we march along a chromosome, many genotypes at particular regions of the chromosome are not going to show an association with the phenotype, in this case, some aspect of parental behavior. But what we're looking for are regions of the genome in which genotype, that is, whether they have two alleles from a monogamous versus a promiscuous parent, are predictive of the behavior of interest. And so what that will look like is that in this particular region, we'll see an increase in a LOD score, which you can think of as the genetic association between genotype and phenotype, and suggests somewhere in this region, when it passes genome-wide significance, there's likely to be a variant or variants that contribute to the trait of interest. Okay, so next what I want to do is to show you some, in some sense, raw data from this QTL analysis to give you a sense of the data, and then I'm going to summarize that data. So when we're looking at QTL for parental behavior, the first example I want to show you is of nest quality. So here we're marching across chromosomes or linkage groups. I'm showing you data again for mothers and fathers separately. And what you can see is in two regions of the genome, in linkage group 3 and linkage group 19, you can see our LOD scores pass our genome-wide significant threshold, and does so for both males and females. So that suggests that there's a variant somewhere on chromosome 3 and chromosome 19 that contribute to differences in nest building behavior between the species that affects both males and females. Other traits look somewhat different. So here we're looking at the latency to handle pups. And what you can see here is that on a different chromosome, there's an association, but that association is just for fathers. So only explains differences in latency to handle pups in males. Okay, so next I'm going to take all of this type of data and summarize it in this graph. So what you're looking at here are 6 focal behaviors, again, going chromosomes across the horizontal. Males and females separately, and the intensity of the color gives you a sense of the strength of association between genotype and phenotype. And those that are significant are boxed out with this black outline. So the first thing you might notice is that there's actually several regions across the genome that affect parental behavior. But what's more interesting is when we hone in on particular genetic regions. So for example, this particular region on chromosome 12 seems to be associated with variation in multiple behaviors. So licking, huddling, retrieving, for example. So a little bit of a behavioral module. Whereas other regions like this one on chromosome 4 seems very specific to, in this case, nest building behavior. Some of these regions like this one affect both male and female behavior, whereas others like this one on chromosome 5 seems to be male specific. So what we're seeing when we look at the genetic architecture is consistent with the data I showed you when we looked at correlations among behaviors. That is, some behaviors are highly correlated, likely caused by shared genetic variants, whereas others seem to be specific, and some are sex specific and some are not. Okay, so now here's the part where it's hard to be a geneticist, because we found these associations, but the associated genomic regions are actually quite large, often encompassing hundreds of genes or big chunks of chromosomes. And what we really want to do is narrow in within those regions to identify some candidate genes, and just a handful, enough that we can, not so many that we can't functionally test them. So there are many ways in which you can try to narrow those potential genes into a handful of candidate genes. And in this case, we decided to complement our QTL approach with comparative RNA-Seq. So what we were interested in doing in this case was we focused on one region of the brain, the hypothalamus, because the hypothalamus has been well established to play a role in innate behaviors and parental care in particular. So we dissected out the hypothalamus of both males and females from the two species and conducted, in this case, just bulk RNA-Seq and asked which genes are differentially expressed. That on its own is not very informative, but when we intersect that with the genes that fall within our QTL peaks, we can start to narrow our list of candidate genes with the assumption that the causal change affects gene expression. So here I'm going to start to focus now on one QTL peak in particular, and that's the one that affects nest quality. And I just wanted to remind you about this particular QTL, sort of two facts that are going to become important later. First, just by way of reminder, this region of the genome seems to be associated with variation in nest building behavior in both males and females, and it seems to be specific to nest building and not the other aspects of parental behavior we measured. So this particular QTL encompasses about 500 protein coding genes. And as I mentioned, when we do RNA-Seq expression, we see lots of differences between these two species, even though they're only recently diverged. But what we're most interested in is asking, of the genes that fall under the QTL peak, how many of those show differences in gene expression? And it turns out there are 23 protein coding genes. So this is interesting, but still a pretty large list to start to functionally perturb. So we did one more step to narrow our list of candidate genes or prioritize them. And that is, we dissected out the hypothalamus of an F1 hybrid and asked, are there differences in the expression of the two alleles in this F1 hybrid, one from the monogamous parent, one from the promiscuous parent, in a common environment? When we did that, we saw that of those 23 genes, 9 of them showed allele-specific expression differences. And I've ordered them here by fold difference across the horizontal. And of these genes, one gene in particular sort of stood out to us, just at quick glance, and it may catch your eye too, and that's this gene called gene 1. So gene 1 shows the largest difference in expression between the promiscuous and monogamous species. And it's pretty robustly expressed in the hypothalamus, which is not a big indication that it's a great candidate, but consistent with it maybe. And this has got pretty exciting because this suggests that this gene has a difference in the cis regulation of this gene, which is consistent with it being under our QTL peak as opposed to showing a difference in gene expression because of a trans-acting factor. So this gene got us really excited, and even more excited when we learned what this gene was. It turns out this gene encodes for arginine vasopressin. And I already hinted at the beginning of the talk that we know from studies in mice, and even more so in voles, that the oxytocin vasopressin system has been important in variation in social behavior. So it turns out that vasopressin is expressed at higher levels in promiscuous mice, so threefold higher in promiscuous mice than monogamous mice. We saw that in the bulk RNA-Seq. And then when we looked at this allele-specific expression difference, we found a similar expression difference and in the same direction, with the promiscuous allele driving stronger expression than the monogamous allele. So one thing that's important to keep in mind is that in this context, vasopressin seems to act as a negative regulator of nest building behavior. So the higher expression of vasopressin, the lower or the less nest building behavior you display. Okay, so keep that in mind. So this was really exciting. We've narrowed down to this exciting candidate gene, but everything I've shown you so far is just sort of a statistical correlation. The next thing we wanted to do is ask, if we perturb vasopressin expression, do we see an effect on nest building behavior? And we thought really hard about what kind of experiment would be the most convincing, what would really convince us that this gene was contributing to differences in nest building behavior. And so we were very lucky because down the hall and around the corner, my colleague Catherine Dulac has her lab. And she happened to have an AVP Cree mouse that one of her postdocs had generated available for us to use. So this would enable us to test the role of differences in AVP in laboratory mice. So we designed a chemogenetic experiment using DREAD receptors. And as I'm sure many of you know, DREAD receptors are designer receptors that allow us to increase or decrease neuronal activity. In this case, we were interested in vasopressin expressing neurons. So we can express these DREAD receptors either to activate or decrease vasopressin. And we could do that both in a very temporal and spatial specific way. Temporal in the sense that we can activate these designer receptors with a designer ligand, CNO, which is designed just to interact with DREAD receptor and hopefully nothing else in the body of the mouse. And we can do this in a very spatially specific way because we could inject this construct into just a particular nucleus of the hypothalamus. We focused on the paraventricular nucleus because it had the highest difference in expression of vasopressin compared to other nuclei in the hypothalamus. And here you can see using M-cherry as a marker of our DREAD receptor that we can get really nice spatially specific expression of this receptor in the PVN, but not neighboring nuclei. So this experimental setup then allowed us to test the role of AVP in nest building behavior in these laboratory mice. So we got this whole system set up and we thought, okay, the first thing we want to do, let's inhibit vasopressin expressing neurons and let's start with males. So we did this experiment, held our breath, and lo and behold, we saw a difference compared to controls. When we inhibited vasopressin expressing neurons, we saw an increase in nest building behavior. So this is exactly what we sort of expected. And I have to say, in retrospect, maybe we should have stopped the experiment here, but instead we decided to continue on and test this in females. Unlike males, when we did this experiment in females, but maybe we saw a trend, we definitely didn't see any significant effect on nest building behavior. So we sort of scratched our head and said, okay, well, let's try to activate vasopressin expressing neurons. We started with males. No effect. But when we repeated the experiment in females, we saw an effect and in the right direction, that when we increase vasopressin, we decrease nest building behavior. Okay, so we sort of scratched our head at this result and said, well, maybe we don't recapitulate perfectly what we expect in Perimiscus, that it affects both males and females. And in both of these cases, in part because we're doing this in a species, as I told you earlier, that's separated by 25 million years of evolution. And then we also reminded ourselves that AVP neurons tend to be sexually dimorphic, and they tend to be in general in rodents, and they tend to be more numerous in males, which may explain why in this experimental context, we could inhibit in males and activate in females, but didn't have the power to do the reverse. So we were pretty convinced that that was a good explanation. So we decided to write up this paper and send it in for review. And here's where I'm sure all of you can empathize to some extent, because we waited nervously for months, got the reviews back. And of course, we had, like many, as we had in the past and many of you experienced, sort of dreaded reviewer 3. And reviewer 3 was very complimentary. So I hope it's none of you in the audience, very complimentary, but pointed out that really what we wanted to do, if we wanted to do this experiment right, was to manipulate AVP expression, not in laboratory mice, but in Perimiscus. And we completely agree. We would love to have an AVP Cree mouse Perimiscus mouse in the lab and repeat these experiments in Perimiscus. At the time, we didn't even have transgenesis up and running for Perimiscus. So we thought after reading this one review, well, maybe the editor might be sympathetic to the fact we're working in a non-model organism and we just don't have the genetic tools available. But it turns out reviewer 3 was not the only one to make this comment. So was reviewer 4 and reviewer 2. So this really convinced us that we needed to attempt another type of functional experiment to see if we could get results more consistent with the behavioral differences we were seeing in Perimiscus. So we spent a lot of time sort of thinking about what could this be, and we ended up returning to an idea that was the first idea that we had, and that is just doing simple pharmacology. What if we can just inject AVP into the brain, into an intercerebellar ventricle of the brain of Polyunos and see if it affects nest building behavior? So why didn't we do this initially? We thought, that would be a great experiment, but it'll never work. It'll never work because vasopressin is such a highly pleiotropic gene, right? It affects osmoregulation, blood pressure, et cetera. Like if we, inject this into the brain, we're never going to see, we might see an effect, but it's going to be just very generalized, and it's not going to tell us very much. But bolstered by these reviews, we thought we should at least give it a try. And at least we could go back to the editor and say, we tried. So we set up this experiment again with help from Catherine Dulac, and we focused on Polyunos. Remember the mice that build nice big nests? So if we inject arginine vasopressin, and the expectation would be that their nest building behavior goes down. So when we did this experiment, we were sort of fell out of our chairs when we saw that, in fact, that's exactly what we saw from this sort of relatively simple pharmacological experiment. So compared to controls, either Ringer solution, or we also used oxytocin as a control because there's crosstalk between vasopressin and oxytocin pathways. But when we injected vasopressin, what we saw was a difference in nest building behavior, and in the right direction, it went down. And it turns out this is even more interesting because it didn't just happen in males, it happened in males and females, as was predicted by the genetics. Then we could also do another experiment, because the other prediction from the genetics was that it affected males and females, but also it was specific to nest building and not the other parental behaviors that we measured. So in these same mice, we measured other aspects of parental care and found that, as predicted, there was no effect of vasopressin on huddling, licking, retrieving, or any other behavior we measured. It had a specific effect on nest building behavior. So this was really exciting because it matched the predictions based on the genetics. But what was even more interesting was that at the time I had a PhD student, Caitlin Luarch, who was also studying nest building behavior, but she was studying nest building behavior outside a parental context and was focusing on virgin animals. And even in virgin animals, there were big differences in nest building behavior between Polyunos and Maniculatus, with again, the monogamous species being better nest builders in a given amount of time. So she also did a QTL study and asked, are there regions of the genome associated with this difference? And she too, thankfully, found some nice QTL peaks. But what was especially interesting about this genetic architecture is she didn't find a QTL peak on chromosome 4, which is where AVP sits. So the implication of that is that vasopressin has a specific effect on nest building behavior, but only in the context of being parents. We don't see a same effect when the mice are virgins. So the effect is even more specific. And just as a side note, we also found a different QTL on chromosome 9 that was associated with differences in nest building behavior in virgin animals, and have now narrowed down onto some exciting candidate genes that have not been associated with nest building behavior previously, or any behaviors for that matter. Okay, so here I want to take a step back and summarize what I've told you. So this study really started with this natural history observation that these two species had different mating systems. By bringing these mice into the lab, we were able to show that those differences in mating systems translated to differences in parental behavior, and that those differences appeared to be heritable. Then using forward genetics, we were able to narrow in on particular regions of the genome that contain causal variants that affected nest building behavior. And by intersecting that forward genetic data with RNA-seq data from the hypothalamus, we could implicate AVP as a strong candidate gene where expression differences were correlated with differences in behavior. And then using chemogenetics in mus and pharmacology in Perimiscus, we could show that there was a direct link between expression levels of AVP and behavioral variation. So for us, this was a really nice demonstration that even in non-model species, we're able to go from strong heritable differences in behavior down to the underlying genetic variation. So what did we learn from this study? So one of the surprises was that males and females can evolve similar parental behaviors via different genetic mechanisms. And that's as indicated because some of these QTLs seem to have very sex-specific effects. We also learned that some genetic variants can affect multiple behaviors, whereas others can have very specific effects. And in particular, AVP is one of these that seem to have a very specific effect because it was associated only with differences in nest building behavior, not other aspects of parental behavior, and only nest building behavior in the context of being a parent, not in virgin animals. And this is actually quite surprising because, as I mentioned earlier, vasopressin is a highly pleiotropic neuropeptide. And while the vasopressin and oxytocin pathways had been previously sort of associated with differences in social behavior, the hypothesis really was that most of the action was going to be in the receptors, in the vasopressin or oxytocin receptors, because you can tweak where those are expressed in the brain to potentially produce very specific effects on behavior. Whereas the vasopressin ligand, on the other hand, because it's so highly pleiotropic, the idea was you don't want to mess with the ligands in an evolutionary sense because you're going to have very general effects on behavior. But that turned out to be not the case. So in fact, vasopressin wasn't a standout candidate gene because of its highly pleiotropic nature. But nonetheless, because we used an unbiased genome-wide forward screen, we could really implicate a gene that, at least to us initially, wasn't a prime candidate gene. And I've hoped to at least hint the fact that as we move forward with projects like this one here, we'll move away from really well-established, in this case, neuropeptides, to start to uncover truly novel genes and novel mechanisms. So as I mentioned at the very beginning of the talk, the question we were interested in are what are the genetic changes responsible for natural variation? And here I gave you an example of behavior where we were able to spend a lot of time and energy identifying a gene. And as I mentioned, now is where for us a lot of the fun work starts, because we can start to use this gene as a handle to try to understand the evolutionary processes and molecular mechanisms that give rise to this variation in social behavior. So for example, we're looking at signatures of selection using population genetic approach in and around vasopressin. We're asking questions about convergent evolution. Is vasopressin also acting in the other part of the Perimiscus clade associated with monogamous species and differences in parental behavior? And we're also very interested in what are the precise mutations around vasopressin that lead to these differences in expression, as well as the underlying neural mechanisms that give rise to these differences. So trying to understand not only differences, if for example, we're seeing that there are very big differences in the number of vasopressin expressing neurons between species, and asking if this is contributing to differences in gene expression. So while a major goal of neuroscience, I think in general neurobiology, is to try to understand how genes act in or on neural circuits to produce behaviors, our lab is really trying to capitalize on variation in genes to understand how they contribute to variation in behavior, and then using those genes to ultimately understand how variation in neural circuits give rise to those differences in behavior. And with that, let me end by thanking the collaborators on this project, in particular, Andres Bendeski, shown here in the middle, who was really the main driver of this project. He's now started his own lab at Columbia at the Zuckerman Institute, where he's continuing some of this work. And Catherine Dulac's lab, again, luckily down the hall. And I mentioned some of Caitlin Luarch's work on nest building behavior in virgin animals. And with that, I'm happy to answer any questions. Thank you so much for your attention.
So Hopi, that was fantastic. And as a geneticist, I just love, that you're able to connect variation to such fascinating behavior and, in such a clear way. There are a few questions that people already have. Please put your questions in the Q&A, but I just wanted to ask one first, which is, to go back to your result with the virgin animals, where you don't see the effect of the vasopressin ligand. I guess as a general question, you might say that it was pretty lucky that everything worked out so well in the example of the parenting. And I just wonder, do you think, has it got to the point where you can really sort of reliably count on getting from a phenotype that you think is heritable to the contributory variation in a quote unquote non-model organism for all the things you're interested in? Where you don't have something like obvious like vasopressin to stand out.
Yeah, and I think that's a good question. And I think a lot about that term lucky, in part because I do feel like sometimes we get really lucky. But I think that we try to maximize our chances of being lucky. And by that, what I mean by that is that we choose phenotypes to study very carefully. So for example, we really spent a lot of time thinking about what, in this case, behaviors we could measure reliably and repeatably and accurately in a high throughput enough way that we could get good sample sizes. We try to choose behaviors that are really different, so just maximizing our power. We choose behaviors that are, again, highly divergent, but between closely related species. And that, I think, is really important because the species in this case, let's say, are separated by about 1 million years. There's just not that much evolutionary time to accumulate hundreds of mutations, but instead probably fewer mutations. And if the differences are really big, maybe they are large effect mutations, which of course are easier to find. And so in that sense, I think we try to maximize the chances that we'll find something. We do have examples in which either we find nothing, no peaks, but that's been actually pretty rare. Most often we find peaks, in part, I think, because we set ourselves up to find peaks by choosing carefully. But what really it comes down to is that in some cases within those QTL peaks, there's something to go after. And there are other peaks where there's nothing obvious. And this is why I sort of tried to emphasize this point in the beginning when I was talking about pigmentation. The first few examples of finding genes associated with pigmentations were the candidates. They were MC1R and agouti. But then as we dug in more and our sample sizes were bigger and we did more fine scale mapping, that's when we finally got to the point where we were identifying genes that weren't even known to be involved in pigmentation. So I think that we're just entering that stage with behavior where we're starting to think about like ways in which we can go after genes that are not obvious. And we're taking a multifaceted approach. So here was just an example where we use bulk RNA-seq. We are now, the tools are more available. There are more omics approaches, for example, but there's also other approaches that we can now apply to non-model systems that are really helping us. So I will say that in my optimistic view, yes, we're at the point we can do this. We're not always going to be able to do it, and it's not going to be easy. But I think that if you choose wisely and carefully, it's certainly possible. And this just being maybe one of the first examples in which you can really do that. But I think hopefully really inspires people to try, because it's possible, in my opinion.
So we have a few other questions. Hopefully we can get through all of them. So here's a question from Ann Churchland, which is, did you have any thoughts on why the DREAD results were so puzzling?
Yeah, I think, okay, the two main things, which are the two obvious things, is one, we did this in mus. And 25 million years of evolution between the two, you just don't know what's different. So I think that could be one. The second thing, I think that this sexual dimorphism, the idea that males have generally more of these vasopressin expressing neurons than females, and we've now shown that that's actually true in Perimiscus. I can't remember how much we know about that in mus, but it's true for rats, if I remember correctly. Makes it at least plausible that if you have a lot in males, then you can inhibit them quite nicely or with some power. And if you don't have many in females, activating them can give you an effect, but maybe not in those other two corners. That's a little bit hand wavy, I think. I almost buy the idea that like just doing something in a different species is just really hard to recapitulate the results so precisely. So I think that, and then, of course, there's the DREAD system itself, right? There's some issues with that, and their sample sizes weren't huge and so forth. It's a pretty rough behavior. We're sort of scoring nest building in 30 minutes. So maybe if we broke down that behavior into more precise modules, we might be able to detect smaller effects. So I think there's just a lot of explanations, and I don't know. That's my best guess, I would say.
Okay, we have a question from Larry Zapersky, who wants to know, out of curiosity, where are the receptors for AVP expressed?
Oh, that's a great question. And we've started to look at this very carefully in Perimiscus. I'll have to get back to you on that. So we see these vasopressin expressing neurons obviously in the hypothalamus, but if you're asking where they extend to, yep, I don't know off the top of my head, but I can find that out if I look at my notes.
Okay, great. Okay. Here's a question from Ann Andrews. Your QTL in virgins suggested that AVP is important in nest building during parenting. Did you test this by injecting AVP intraventricularly in virgins? And also, which strain of laboratory mice did you use in the AVP injection experiments?
Okay, so the second question, the chemogenetic experiments, we used C57 black. That's what Catherine had in her lab. And we didn't try other strains. In terms of, did we try injecting AVP in virgin Perimiscus? We didn't. It would be really interesting to have a look at that. But I'll just add as a note that these experiments were very difficult, even though you're just, in some sense, you're just injecting. Our wild Perimiscus do not like having cannulas on their heads. I'm not sure anybody does, but they didn't like them at all and chewed them off and all sorts of problems. So I think that's the real reason we didn't do it right away. But I think it would be worth thinking about doing it now if we wanted to spend a little bit more time digging into that virgin result. It's a great idea.
Okay, and then finally, one last question from Wei Zhang, which is, he was curious if you measured other behaviors like stress or anxiety related behaviors that might indirectly influence the display of parental behavior.
Yep. So we, in the same cross, one of the things we looked at was exploratory behavior. We ran them in an elevated plus maze, which, depending on who you talk to, whether or not that's an appropriate measure of anxiety. And we did see differences with Perimiscus maniculatus. The worst parenter is definitely more anxious. We have mapped those differences in anxiety as measured through this elevated plus maze, and they don't map to chromosome 4, the region of chromosome 4 that contains AVP. So at least that in that simple analysis, there's not a direct link between anxiety levels and the AVP part of these parental differences. But we are, with Andres, who has sort of taken that project with him to Columbia, are starting to look at the genetic basis of these differences in performance in the elevated plus maze. So stay tuned. Interestingly, there is a QTL for anxiety on chromosome 9 that overlaps with the virgin nest building behavior. Whether or not that's biologically meaningful or not, we don't know yet. That's a great question. You sound like a reviewer, one of our reviewers. Not reviewer 3.
Reviewer 3. Okay, well, anyway, Hopi, thanks so much for a really fascinating talk. And again, we'll look forward to seeing you in person in the future. So, and thanks everybody for being here.
Thank you. Thank you for hosting me.